Here in Utah, where life is elevated, 800 feet above downtown Salt Lake, the Hill Lab at the University of Utah is hard at work. We're interested in studying the parts of biology so tiny that we can't even see them with a regular microscope. Thanks to developments in scientific technology, we can capture micrograph images of tiny biomolecules like proteins. We use structural methods to reconstruct a 3D image from the 2D data collected, getting resolution so high that we can determine the physical structure of the protein, often down to the specific arrangement and type of atoms. But why care so much about these little details? Changes to tiny parts of a tiny protein can have big effects. Proteins are made from combinations of 20 different amino acid building blocks. Each unique protein sequence folds into thousands of different structures. These different structures lead to different properties, which gives each protein a different function. Proteins are workhorses in our bodies, with jobs ranging from signaling hunger to sensing light to warming us up in the late cold night. With structural methods, we can understand how changes to a protein structure or loss of important parts might lead to a change in protein function, as can occur with regulatory proteins in cancer cells. We also might see a protein change into a different conformation when it binds to a certain protein partner or drug. For example, the insulin receptor is an important protein for energy regulation. It nestles in the surface of cells in our body and can sense the signal insulin when it binds. The Hill Lab wants to know exactly how this receptor physically senses insulin and sparks the cascade of messages required to bring sugar into our cells to give us energy. What happens to the signal if we tweak the insulin molecule? Does it still bind the insulin receptor? Does it work longer? Is it inhibited? Answers to these questions are important to inform engineering of insulin drugs and help people with diabetes take up exactly the range of sugar that their bodies need to survive. So there you have it. Proteins have different functions and interactions depending on their structures. The Hill Lab resolves different protein structures and characterizes their biochemical properties, and this gives us insight we need for advances in human medicine. Want to learn more? We'd love to share with you. Check us out online at the University of Utah Department of Biochemistry website, where you can see details of our past and present work and see what other biochemistry labs are doing too.